Namaskar. Today we'll study beta-1 adrenergic receptor, where both adrenaline and noradrenaline can act as agonists. So beta-1 adrenergic receptor is a G protein coupled receptor, a type of GS. So S for stimulatory, and the effector is adenylate cyclase, so it will activate adenyl cyclase. And as we know, it is a G protein coupled receptor and it is a type of GS type, so the alpha subunit alpha S activates adenylate cyclase, and by activating the adenylate cyclase, the second messenger cyclic AMP is produced. For details, you can refer to our earlier classes on G protein coupled receptor and the adenylate cyclase cyclic AMP pathway of G protein coupled receptor also you can see. So there will be production of cyclic AMP, and this cyclic AMP will activate the protein kinase A. So protein kinase A in its inactive form has two regulatory subunits and two catalytic subunits. Cyclic AMP, when binds to the two regulatory subunits at four sites, the catalytic subunits are released, which is the active protein kinase A. And this active protein kinase A can activate troponin, the regulatory protein in cardiac muscle, as well as it will activate the ryanodine receptor to release calcium, and calcium by binding to troponin also activates troponin. So you can see the cardiac muscle. In the cardiac muscle there is thick filament and thin filament. It is similar to the skeletal muscle, and the mechanism of cardiac muscle contraction is slide filament theory, similar to skeletal muscles, and these thin filaments are slided towards the thick filament when there will be actin myosin binding. Thick filament has the myosin and the contractile protein, and thin filament has the contractile protein actin. When actin myosin binding occurs, the thin filament slide towards the thick filament. So this myosin forms this thick filament, where the myosin tail forms the thick filament and the heads are projected upwards, and these heads can bind to actin in the thin filament, and this is the structure of a thin filament, and this is the structure of actin. Actin has a site called myosin binding site to which myosin head can bind, and these myosin binding sites of actin are covered by a regulatory protein called tropomyosin, and tropomyosin is again regulated by troponin. So two regulatory proteins, troponin, tropomyosin, two contractile proteins, actin and myosin, are there in cardiac muscle. So calcium can bind to this troponin and replace the tropomyosin, so that the myosin binding sites of actin will be exposed, and myosin heads will now bind to the actin, and they will pull the thin filament towards the thick filament, and because of that there will be sliding of thin filaments, and there will be contraction of the Z disc, and there will be that will result in contraction of cardiac muscles. So protein kinase A, active protein kinase A, can activate troponin and can also release calcium, which will bind to the troponin, and now this active troponin will alter tropomyosin, so that actin myosin binding can occur and contraction of cardiac muscles occur. Now because of this there will be increase in heart rate, increase in stroke volume because of myocardial contractility. So, and in kidney there is also renin activity because of beta-1 receptor. In heart there will be increase in heart rate and increase in stroke volume, and as you know, increase in heart rate and increase in stroke volume will result in increase in cardiac output, because heart rate into stroke volume is cardiac output. And that means stroke volume is the volume ejected, volume of blood ejected by heart per stroke. So into number of stroke or number heart rate will give us the volume of blood ejected per minute, that is cardiac output, and cardiac output into peripheral resistance is blood pressure. So by increasing heart rate and increasing stroke volume, cardiac output increases and hence blood pressure will also increase. So we use beta blockers like atenolol, metoprolol as antihypertensives. In other cardiac diseases, to decrease the workload of heart, we can use beta-1 blockers. For example, in angina pectoris, there is myocardial ischemia, so we need to decrease the workload. In that case, we can use the beta-1 blockers, which will decrease the cardiac output, hence the workload of heart will decrease. And beta-1 agonist example is dobutamine. So, and beta-1 receptors not only in heart, in kidney also, by increasing the renin activity, the renin-angiotensin system is activated. We call it as RAS, renin-angiotensin system, where there will be production of two components. One is aldosterone. Aldosterone causes salt and water retention, and the other is angiotensin 2, which acts on AT1 receptor to cause vasoconstriction. So AT1 receptor in blood vessels causes vasoconstriction, and peripheral resistance increases. So in this way also blood pressure increases, and hence we can use the beta-1 blockers as antihypertensives. So this is all about the beta-1 receptor, its mechanism, the pathway, exact pathway, how the second messenger cyclic AMP functions. So this is all about beta-1 receptor. Thank you.